Hello everyone, welcome back to our YouTube channel AgriAddict. As you already know by now, that AgriAddict has launched its free lecture series for the IBPS A4 mains exam, and today our topic of discussion is plant disease management. Myself Ushasi Roy, I have completed my undergraduation from Bidhan Chandra Krishi Vishwavidyalaya, Mohanpur, West Bengal, and currently I am pursuing my masters in the Department of Plant Pathology at University of Agricultural Sciences, Harvard. So today in this video, we would like to talk about the following topics. Firstly, we will talk about the features of the plant disease management, and we will also learn about the principal methods that we use to manage a plant disease efficiently. So the first thing that comes in our mind is, why do we need to manage a plant disease? We know that when a disease occurs to a certain plant, it reduces the production capacity of that plant. So we need to control the spread of the plant pathogen, that is, the causal organism of this disease. We need to remove this hindrance so that we can attain a maximum production capacity of that plant. So for this reason, we need a proper management strategy. This management strategy includes various methods like cultural methods, chemical methods. Even the farmer can adopt the cultivation of disease-resistant varieties. So all this comes under the management strategy. The plant disease management aims at the eradication of the pathogen, that is, the removal of the source of infection or inoculum. It also helps in maintaining the damage below an economic injury level so that the farmer doesn't face an economic loss. It also aims at minimizing the occurrence of the disease. So, what are the features of the plant disease management? It should be cost effective. That is, the method that the farmer is adopting to manage a plant disease, the cost of it should be less than the loss he will incur if the disease occurs to the plant. So that it should be cost effective in nature and it should be feasible. It need to be integrated in approach. We know that a plant is affected by various external factors like weed, insect, and disease. All these three things together affect the production of the plant. So we need to adopt the method by adopting which all these three things can be managed together so that it should be integrated in approach and it should be scheduled according to the particular growth stage of the crop. That is, the method is specific to that growth stage. The management of the surroundings of the crop field is necessary. That is, we need to manage the inoculum, the source of infection, and vector. Vector is the agent that helps in the dispersion of the plant pathogen from the infected plant to the healthy plant. So we need to manage the vector properly. So to devise a proper management strategy, we need to know the disease triangle. The manipulation of the disease triangle is necessary. Then only we can manage a disease properly. So what is disease triangle? It comprises of host, pathogen, and the environment. The host should be susceptible to the pathogen. The pathogen should be virulent and the environment should be favorable. When all these three things interact together, it helps in the occurrence of the disease. So we need to manipulate the disease triangle. That means we need to prevent the interaction between the host and the pathogen and the environment, and between the host and the environment. So we need to manage the host. The modification of the environment is necessary, and we also need to manage the pathogen. Host management can be done by the protection of the planting material. The planting material means seed or any propagating materials like tuber, bulbs. So those things we need to procure from the disease-free areas. Prevention of the post-harvest decay, that is after the harvest, we need to protect the post-harvested produce from various diseases. Protection of the foliage by the application of the chemicals. Cross protection. This method is very much useful in managing a viral disease. So here what happens? The mild strain of the virus is artificially inoculated in the plant so that it develops a resistance, and in later stage it helps the plant to prevent the attack from the severe strains of that virus. Okay. We also need to induce the resistance in the plant. Various plant breeding methods for disease resistance are there. That is, we need to exploit the horizontal and the vertical resistance already present in the germplasm. And we also need to adopt various biotechnological approaches, that is transgenes and genetic manipulation. These 3 points will be discussed later in the slides. Pathogen management. So how do we need to manage the pathogen? For this reason, we need to have a clear knowledge about the disease cycle of that pathogen so that we can break this infection chain. We can manipulate it. We need to avoid the contact between the host and the pathogen. We need to reduce the inoculum potential by sanitation of the host, that is protection of the plant and the environment. Prevention of the infection by the adoption of various chemicals. That is, we need to apply various chemicals like the fungicides and the use of the biocontrol agents. The environment modification is also necessary, and we need to modify environment in such a way that it does not affect the plant growth, but it affects the life cycle of the pathogen. That is, it disrupts this infection chain. It can be done by crop rotation, soil solarization, and nutrient management. That is the control application of various nutrients like NPK, nitrogen, phosphorus, and potash. The control application and their proper dose can help in the prevention of the occurrence of the disease. The convention approach to the plant disease management revolves around immunization and the prophylaxis system. So, what do you mean by immunization? It refers to the induction of the resistance in the host plant so that the host, that is the plant, can resist the attack of the pathogen. And what is prophylaxis? So, prophylaxis refers to the preventive measures. Okay, so prophylaxis system refers to the disease occurrence prevention. So, there are various principal methods by which we can control a plant disease. Avoidance, exclusion, eradication, protection, resistance, and therapy. These are the principal methods that we adopt for managing a plant disease. We will talk about each of them. So, firstly, it comes avoidance. So, from the word avoidance, we can have an idea that we need to avoid making contact between the host and the pathogen, and it ensures that the susceptible stage of the plant and the favorable environment conditions for the proliferation of pathogen does not coincide. This avoidance principle is adopted before the onset of the disease, that is, it is prophylactic in nature. The methods that come under avoidances are: we need to select a proper geographic area so that the disease doesn't occur in that area. For example, ergot of Bajra. This is a disease caused by Claviceps fusiformis. It is very much severe in wet areas. So, we need to cultivate Bajra preferably in the dry area to avoid this disease occurrence. We need to select our field properly. It can be done by judging the field in the parameters like drainage condition of the field. Red rot of sugarcane is very much prevalent in the flooded areas where the water stagnation occurs. So we need to avoid it. And also the inoculum potential of various soil-borne pathogens. There are various soil-borne pathogens like fungus, we have Pythium, Phytophthora, and various bacterial diseases. For example, bacterial wilt of solanaceous plants. So we need to choose a field where the bacterial wilt of solanaceous plants is not prevalent. That is, the inoculum of that pathogen is absent in that field. We need to have the proper schedule of the time of sowing of that particular crop. That is, delay in sowing in case of gram to avoid the rhizoctonia root rot. Various disease escaping varieties are also there. We need to cultivate them. So the disease escaping varieties, it is not the genetic resistance, okay, but it is the characteristics of the growth and the time of the maturity of the plant. For example, the early maturing varieties of pea, it can escape the powdery mildew. We need to select the seed and the planting stock from the areas where it is disease free. And the example is I have given that the seed potato should be procured from Shimla, so that the southern areas they generally procure the seed potato from Shimla to avoid the occurrence of various potato related diseases, that is lead blight and various other viral diseases. We need to modify the cultural practices. For example, tillage. It is the effective method of the control of the pathogens, because we know that tillage disturbs the soil and it also destroys the crop residues. So the inoculum potential that is already in the crop residue, it gets destroyed. The distance between the plants also matters. Like in case of seedlings, if they are close planted, it results in the occurrence of damping off, so that the proper distance between the plants should be maintained. Flooding. It helps to get rid of the sclerotia. It is the resting body or the resting spore of the sclerotinia pathogen. The flooding helps in the removal of the sclerotia from the soil. Irrigation methods. The sprinkler irrigation is avoided because it helps in the dispersion of various bacteria. Bacterial disease, it preferably disperses. So the sprinkler irrigation is avoided. Rather, controlled irrigation is adopted. And it is generally applied for various soil-borne pathogens like Pythium and Phytophthora. Mixed cropping can also be done because it checks the spread of the various diseases. Example, cotton and pulse crop. The mixed cropping of this crop should be done and it reduces the incidence of the root rot of cotton. So I hope that this avoidance method is clear to you, and you need to remember that what are the various methods that comes under avoidance. So our next principal method is exclusion. So what do you mean by exclusion? It prevents the entry of the pathogen from the infected area to a new area. New area means it is the area where the disease is not prevalent. So it prevents the entry of the pathogen from the infected area to an uninfected area. And it is also adopted before the onset of the disease. That is, it also helps in the prevention of the occurrence of the disease. It includes various methods like seed treatment. The propagating material are protected by various treatments. The heat can be used, gases can be used, or various chemical treatments. That is, various fungicides are used for the protection of the planting materials. Inspection and certification is done. So, what is inspection? Like periodic inspection of the seeds. Like, for example, during the import of the planting material from abroad to India, the planting material is first inspected. That is, it is examined whether there is the presence of any pathogen or any pest, whether it is present or not. It is examined. So that is the periodic inspection of the seeds for preventing the infection by various seed-borne pathogens. Okay, and if the material is examined and the result comes that no, it is free from all the diseases, then the phytosanitary certificate is issued. That refers that the planting material that has been procured from outside is okay. That is, it is free from the pest and the seed-borne pathogens. Quarantine regulation also comes under exclusion. So, what is quarantine regulation? It is a legal restriction on the movement of the agricultural commodities for the purpose of exclusion, prevention, or delaying the spread of the plant pest and the diseases in the uninfected areas. And that's why the planting materials are held in isolation. Quarantine means to keep the planting materials in isolation. It is kept isolated so that it prevents the entry of the pathogens and the pest from the infected area to the uninfected area. Quarantine can be of three types. It can be of domestic. That means it restricts the entry or the import of materials from one state to another state. For example, in West Bengal, potato wart occurs. Okay. So for this reason, the potato seeds are not imported from West Bengal to the other state because there is a threat that the potato wort can be introduced from West Bengal to the other state. So this is an example of the domestic quarantine. Foreign quarantine means it prevents the entry of the diseases in a country. Like from a particular country, a particular agricultural commodity is not imported to India because of a particular pathogen. And embargo, it refers to the total ban or restriction on the import and the export of the agricultural commodity. There are various points that you need to remember. That is the Plant Quarantine Act of 1912 in USA was the first phytosanitary and the quarantine import regulation. In India, the Destructive Insect and Pest Act was passed in the year 1914. This is the one of the most important points that you need to remember. Quarantine laws were first enacted in France in 1660. It aimed at the rapid destruction and the eradication of Barbary. Barbary is the alternate host of the black rust of wheat. And currently in India, there are 16 quarantine stations that works under DIPA. 8 are at seaports, 6 are at airports, and 2 on land frontiers. Okay. So I gave here the list of the various diseases that have been introduced from other countries. Okay, so you can remember the most important ones. For example, the late blight of potato. The causal organism name is also given. The year of introduction is given. The place from where it is introduced and to the place where it is introduced. Okay. For example, late blight of potato was introduced from South America to Europe. Powdery mildew of grapes was introduced from USA to England. And here you can see that banana bunchy top was introduced in India from Sri Lanka. So you can memorize this list for the purpose of the exam. Here is another list that gives the name of the diseases that are introduced in India from other countries. For example, leaf rust of coffee. It has been introduced from Sri Lanka to India in the year 1879. Paddy blast caused by Pyricularia gricia. It has been introduced from Southeast Asia. So you can have a look to this list and remember the important ones. And here it is the list of the diseases that are under domestic quarantine in India. I have already mentioned about the potato wart, and the area it is restricted to is West Bengal, and the place name is Darjeeling. Banana bunchy top is the disease that is restricted to areas like Assam, Kerala, Orissa, and West Bengal. Banana mosaic is the disease that is restricted under the area Gujarat and Maharashtra, and onion smut is restricted under Mysore. So I hope that exclusion method is clear to you. The next principal method that we learn about is eradication. So what is eradication? It is the elimination of the pathogen from the infected areas. That is, the pathogen is already present in an area and it is an infected area. So we need to remove that pathogen from that area. And how it is done? It is aimed at breaking the infection chain of the pathogen. It is also adopted before the onset of the disease. That is, it aims at the prevention of the occurrence of the disease. What are the methods that comes under eradication? The first method is rogging. What do you mean by rogging? Rogging is the removal of the infected plants or plant parts from the fields at the early stage. so that the source of the infection or the inoculum is removed, and it prevents the dissemination, that is the dispersal of that pathogen. For example, in loose mart of wheat, in wheat cultivation, if we see like the ear of wheat affected by loose mart, we completely uproot that plant and remove it from the field so that it prevents the dispersion of that pathogen. It also includes the eradication of the host. So host is of two types, okay? Beside the main host, it is of alternate host and collateral host. Alternate host belongs to the different family than the host of the pathogen. And this alternate host is required by the pathogen to complete its life cycle. So this is the most important point. You need to remember because the questions can come that what is alternate host? It can belong to same family or different family of the host. So you need to remember that alternate host belongs to the different family than the host of the pathogen. And it is essential or required by the pathogen to complete its life cycle. Example, the barberry. It is the alternate host for the wheat stem rust. And what is collateral host? It belongs to the same family as that of the host of the pathogen. Okay. It is required by the pathogen to survive during the off season. That is when the main host of the pathogen is not available, it harbor collateral host to survive during the off season. For example, Alternaria solani, it attacks all the members of the Solanaceae family, like potato, tomato. Okay. Crop rotation. So we know that continuous cultivation of the same crop or the related crop, it leads to the buildup of the inoculum potential of the soil-borne pathogens, and it results in the increase in the intensity of the diseases. For example, in late blight of solanaceous crops, like any solanaceous crop you can consider, like for example, potato. The crop rotation with pulse or other vegetables is suggested because in the next season, if we cultivate any other solanaceous crop other than potato, it will recur the occurrence of the same disease. That is, the late blight of that same late blight will occur to the next crop also if it belongs to the Solanaceae family. Sanitation is also necessary. The pathogens survive or perinate through various dormant structures in the various plant parts, like it resides in the crop residue. So we need to collect and destroy those structures. For example, burning of the diseased stubble or the crop residue is suggested so that we can remove those dormant structures of the plant pathogens. In example, it's given that verticillium wilt of cotton. It is managed by the removal of the crop residues after the cultivation of the cotton to remove the inoculum potential of the causal organism of the verticillium wilt. Various heat and the chemical treatment is also suggested for the managing of the disease. For example, hot air treatment is the temperature is given and the time duration is also given. Sometimes this comes, okay, like this is important. You can remember the temperature and the time duration. Like in sugarcane sets, to control the disease red rot, these methods are followed. Hot air treatment is done at 54 degrees Celsius for 8 hours. Moist hot air treatment is done at 54 degrees Celsius for 4 hours. Hot water treatment is done at 50 degrees Celsius for 2 hours. And aerated steam treatment is done at 50 degrees Celsius for 1 hour. Any one of this method can be adopted to control the red rot disease of sugarcane. The systemic fungicides can also be used, that is a chemical treatment. For example, Vitavax and Plantvax. Vitavax is used for the control of the loose mart of wheat, and Plantvax is used to control the rust of wheat. Okay, so here you can there can be a trick that you can remember, like we use CVSOPR. It is a shortcut. Like C is the chemical group to which it belongs, like carboxy, Vitavax, and S stands for smart and do smart. And OPR. O is the chemical name, that is oxycarboxyl. P stands for Plantvax and R stands for rust. Okay, so this is a trick you can remember so that you will remember correctly that which chemical is used for the smart disease and which chemical is used for the rust disease. You just need to remember the abbreviations CVS and OPR. Soil treatment is also done, and it can be done by dry heat, steam, or hot water. For example, carbon disulfide is used in eliminations of Armillaria melia. It is a pathogen, and formaldehyde is used in managing onion smut disease. Biological eradication is also used. Like till now, we have read about all the uh, cultural methods, right? But use of the biocontrol agents also comes under eradication. The reduction of the inoculum density or the disease-producing activities of a pathogen or parasite in its active or the dormant state is done by one or more microorganisms that can be accomplished naturally or through manipulation of the environment, host, or the antagonist by mass introduction of one or more antagonists. So it refers like the pathogen. This is also a microorganism, right? So this pathogen can also be managed by another microorganism that is known as biocontrol agent, BCA, biocontrol agent. So what are the principal mechanisms that a biocontrol agent follows to control that pathogen? It can follow hyperparasitism. So what do you mean by hyperparasitism? We know that this pathogen is already a parasite of the plant, right? Pathogen is a parasite of the plant. It can't live freely. It needs the host to obtain its nutrition. And the biocontrol agent parasitize this pathogen. So this mechanism, it is known as hyperparasitism. So the pathogen is directly attacked by a specific biocontrol agent, and this biocontrol agent kills the hyphae or its propagules. For example, trichoderma. It is the name of the biocontrol agent, a very important biocontrol agent that is used against rhizoctonia. Okay, like trichoderma, hyperparasitosis, rhizoctonia. It can also follow the method of antibiosis. So, what do you mean by antibiosis? This biocontrol agent, for example, trichoderma, it releases various antibiotics. Okay, that is, it releases secrete one or more compounds with antibiotic activity. That is, it releases antibiotics that hinders the growth of the pathogen. The name of the antibiotic is gliotoxin that is released by trichoderma. There are various other antibiotic names, that is viridin, glycotoxin, and others. You don't need to remember all such names. The biocontrol agent can also follow competition. So what is competition? Competition between the pathogens and the non-pathogens, that is the biocontrol agent for the nutrient resources. That is, the example here is given, that is trichoderma. Trichoderma competes with Pythium, the pathogen, for iron availability. So iron is the nutrient essential. It's a micronutrient essential to the pathogen for its survival. Okay, but here the biocontrol agent competes for the iron availability, and as a result, it hinders the growth of Pythium, and then the Pythium, that is a plant pathogen, dies. It can also be done by the induction of resistance. So resistance in the host can be induced by two methods, or there are two types of resistance. That is systemic acquired resistance and induced systemic resistance. So what is SAR, or systemic acquired resistance? It is mediated by the compound generally called salicylic acid, which is frequently produced followed by pathogen infection, and that leads to the expression of the pathogenesis related proteins, that is PR proteins, PR1, PR2, chitinases, or other proteins, and some peroxidases. These PR proteins can cause lysis of the invading cells, reinforcement of cell membranes to resist infections, or induce localized cell death. So, what happens exactly in SAR, that when a plant gets infected by the pathogen, okay, it leads to the secretion of the compound called salicylic acid. Okay, so this salicylic acid triggers pathogenesis and the production of various PR proteins like PR1, PR2. So these proteins ultimately lead to the lysis, that is the death of the cells that are already invaded by the pathogen. That is, it leads to the death of the infected cells. Okay, and it reinforces the cell membranes to resist infection and induce the localized cell death. So the infected cells dies, and it ultimately leads to the eradication of the pathogen from that plant part. And the other type of resistance is known as induced systemic resistance. It is mediated by jasminic acid or ethylene. So you need to remember which compounds induce which type of resistance. SAR, you can remember from the name SAR. So S stands for salicylic acid, okay. And ISR is induced by jasminic acid and ethylene, which are triggered when the plants are colonized by various plant growth promoting fungi or plant growth promoting rhizobacteria. Okay, so these are the beneficial organisms, and when these organisms occupy a plant, it releases jasminic acid and ethylene that induce a systemic resistance in plant, and that latter helps to resist the attack by the plant pathogen. Okay. So the various biocontrol agents names are given, and the products that are commercialized of those biocontrol agent, that is the name of the products by which it is sold in the market, it is given. You can just have a look at it and you can remember some of the important names. For example, Trichoderma herzianum, it is sold under the names like Root Shield, Binapti, if stop. And for example, Aspergillus niger, it is the product name is Calicena. Bacillus subtilis, it is sold under name Kodiak. So you can from here this list you can just have the important names. Okay, you can memorize some of them. Agrobacterium radiobacter. Okay, it is sold under name Galtron. So I hope in this class, in this today's video, you all had a clear overview and you understood clearly about the introductory part of the plant disease management. And we learned today about the three methods, that is avoidance, exclusion, and eradication. In the next video, we will learn about the other principal methods. And if you're looking for any online course for taking preparation for any exam, you can enroll yourself in AgriAddict. They offer online courses for various exams like Vijeta, the target batch for the year 2025 for IBPS A4 mains exam, Sarvata, the target batch for the year 2025 for ICR AICE PhD exam, Sarva Shrestha, the target batch for 2025 for ASRB NET and SMS STO exam, Shrestha batch for the target batch 2025 for the preparation of ICR AIEEA PG exam, and Samartha, the foundation batch 2026 for the ICR AIEEA PG exam. If you want to enroll in any of the courses, please download our app Agriadic, and you can follow us on Instagram, YouTube, and other social medias. You join Agriadic, and from here you will learn from the best to be the best. And if you have any confusion about today's lecture, you can post or comment in our comment box. Our team will get back to you as soon as possible. Thank you. See you in the next video.